Hello, my name is Johnathan Wong. I'm a research programmer at Dr. Naj Bureau's Bioinformatics Technology Lab at Canada's Michael Smith Genome Sciences Centre at BC Cancer. Today I'll be presenting GoldRush, a de novo assembler for long reads with linear time complexity. In the recent years, long reads have risen to prominence. Compared to their short reads counterpart, they have a lower base accuracy ranging from 87 to 98%. As the name suggests, they have longer read lengths ranging from kilobase pairs to megabase pairs. This means they can span over repeats, which usually leads to highly contiguous assemblies. De novo long-read assemblers uses the overlap layout consensus (OLC) paradigm to assemble long reads. The OLC paradigm consists of 3 steps: overlap, which involves aligning all the reads against each other; layout, which uses the alignment to generate a graph and traversing the graph to generate the sequences; and finally, a consensus is computed on the assembled sequences. The OLC paradigm has a quadratic runtime complexity in its naive implementation. Current state-of-the-art de novo long-read assemblers use a modified OLC algorithm to reduce the time complexity. Redbean, for example, segments reads into 256 base pairs to reduce the dynamic programming matrix. Shasta, on the other hand, clusters reads based on the genomic loci they are sequenced from. These modified OLC algorithms still have a high RAM cost to them, ranging from 300 plus gigabytes to 1 terabyte of RAM to assemble a 67X human individual. Here we present GoldRush, a memory-efficient algorithm with a linear time complexity. GoldRush consists of 4 stages: GoldRush-Path, GoldRush-Edit, Tigment-Long, and GoldRush-Link. The raw long reads are first processed by GoldRush-Path to generate base assembly. The base assembly is then sent to GoldRush-Edit for polishing and removing base errors introduced by the base calling software. The polished assembly is then sent to Tigment-Long for correction and cutting chimeric reads introduced by the sequencing platform. Finally, the corrected and polished assembly is sent to GoldRush-Link for scaffolding and generating the final assembly. We now define the golden path, which is an approximately 1x representation of the target genome. To generate the golden path, GoldRush-Path first initializes a data structure to store the reads in the golden path. This data structure is called the multi-index Bloom filter. GoldRush-Path then loops over the reads, querying each read against the multi-index Bloom filter. If the read is not found, the read is added to the golden path and inserted into the multi-index Bloom filter. If it is found, the read is skipped over and the loop continues. This iteration of reads is how GoldRush achieves a linear time complexity in the number of reads. Here is a demo of how GoldRush-Path works in practice. The first read is queried against the multi-index Bloom filter. It is not found, so it is added to the golden path. The second read is also queried against the multi-index Bloom filter. It is also not found and added to the golden path. Finally, for the third read, we also add it to the golden path, but because there's a substantial overlap, we trim it. For the subsequent reads, all of them are found in the golden path, so they are not added to the golden path. After iterating through all of the reads that are inserted into the multi-index Bloom filter is the golden path. To determine if a read is in the multi-index Bloom filter, we use a read signature called space seed. A space seed is a pattern of 1s and 0s, where 1s are care position and 0s are don't care positions. Space seeds are error tolerant, which makes them useful for ONT reads because they have a high base error. Here is an example of how a space seed works. We have 2 k-mers where the middle 2 bases differ. By using this space seed design, where the zeros correspond to the two differing bases, these two k-mers will be considered the same. Before we discuss the multi-index Bloom filter, we must first review the Bloom filter which the multi-index Bloom filter is based on. A Bloom filter is an array that stores ones or zeros, which translates into presence or absence in bioinformatics. This is a very memory-efficient data structure because it is just a bit vector. But because it is just a bit vector, there is a possibility of false positive. What we mean by false positive is if we were to have two distinct elements with the same hash, if we were to insert the former and query the latter, we would find that the latter is inserted, even though we haven't inserted into it. This can be mitigated by using multiple hash functions. To generate the golden path, GoldRush-Path requires a data structure that can tell the presence of this read signature and where the read signature is coming from. To accomplish this, GoldRush-Path uses the multi-index Bloom filter. The multi-index Bloom filter consists of three data structures: a Bloom filter, a rank array, and an ID array. The rank array associates the presence of a read signature in the Bloom filter with an ID in the ID array. This allows the GoldRush-Path to associate a read signature with an ID, which is essentially which read the read signature is coming from. The combination of the three data structures allows the multi-index Bloom filter to create a smaller ID array than the size of the Bloom filter. This allows the multi-index Bloom filter to achieve a significant savings in memory while maintaining a fast access time. To insert a read into the multi-index Bloom filter, the read is first divided into blocks of length t times b, where t is the length of the tile and b is the number of tiles in the block. All read signatures inside the block are inserted with the same ID. Here is an example of a read signature from block 1 being inserted into the Bloom filter of the multi-index Bloom filter and associated with an ID of 1. All subsequent blocks will have their associated ID incremented by 1. To query a read, we first break the read into tiles. Then for each tile, we query all the read signatures and generate an ID and count table. The ID with the highest count will be the representative ID of that tile. By querying tiles instead of the entire read, GoldRush can associate each region of the read with its own ID, instead of associating the entire read with a single ID. By doing this, GoldRush can obtain a higher granularity mapping. Once we have queried every tile of the read, we compile the results and give each tile a preliminary assignment based on whether or not the representative ID has an associated count greater than a threshold. In this case, the threshold is 10. If a tile is assigned true, then the tile is considered to be found in the golden path. Conversely, if a tile is assigned false, then the tile is not found in the golden path. After some more processing, we determine the query outcome of the read. In the first example here, every tile in the read is given a false assignment. This means that none of the tiles are found in the golden path, and we want to insert the read in its entirety. In the second example here, we see a mixture of true and false assignments. This means a portion of the read is already found in the golden path. In this case, we want to trim the read until there's a true tile overhang left and insert the trimmed read into the golden path. Finally, we have an example where all the tiles within the read are given a true assignment. In this case, the entire read is already inserted into the golden path, and we don't have to insert it anymore, so we skip over the read. After GoldRush-Path has generated the base assembly, we will send the base assembly to GoldRush-Edit for polishing. GoldRush-Edit is adapted from NT-Edit plus Sealer protocol for polishing using long reads. The long reads are first mapped to the golden path reads to generate targeted bloom filters. NT-Edit will then use these targeted bloom filters to remove mismatches and indels. For the complex mismatch and indels that cannot resolve, it will flag them and send to Sealer to resolve using a bidirectional de Bruijn graph approach. After GoldRush-Edit has polished the assembly, it is sent to Tigment Long for correction. Tigment Long is adapted from Tigment for long reads. It can detect and correct misassemblies, but is used in this case to identify and cut chimeric golden path reads at the chimeric breakpoint. After the assembly is polished and corrected, it is sent to GoldRush-Link for scaffolding. GoldRush-Link is NT-Link with features developed with GoldRush in mind and applicable to all assemblies. The long reads and the golden path reads are first hashed into their minimizer form. Minimizers are a reduced representation of the read. The minimizers are then used as evidence for NT-Link joins. NT-Link will then detect if there is any overlap in the NT-Link join. It will then trim the overlap so that there is no base duplication. For NT-Link joins with no overlap, it will attempt to fill the gap using reads that provided evidence for the NT-Link join. Finally, the process is repeated to scaffold any NT-Link joins that were not previously discovered. To benchmark GoldRush, we compared it against 3 state-of-the-art de novo long-read assemblers using 3 different human individuals. To benchmark performance, we compared the peak memory usage, measured in gigabytes, against time spent assembling, measured in hours. For this comparison, the lower left corner is where the assemblers want to be at. Here we can see that GoldRush uses a fraction of the RAM that its comparators use and assembles all 3 human individuals within a day. To benchmark contiguity, we ran QoS on all the assemblies using both the GRCH38 human reference genome and the telomere-to-telomere assembly to obtain the NGA50 length and the number of misassemblies. The NGA50 length is the length where at least half the assembly size is in pieces of at least that length, corrected for genome size and alignment block. For both comparisons, the top left corner is where the assemblies want to be at. Here we can see that GoldRush produces assemblies in the 20 meg NGA50 length range with fewer or comparable number of misassemblies to the best assemblers. In summary, we present to the community a novel linear time de novo long read assembly algorithm with a small memory footprint that can assemble human genomes at a high contiguity with low misassemblies. GoldRush is also modular by design. For example, if a user wishes, he can substitute GoldRush-Edit out for any other polishers. I would like to thank my esteemed colleagues for their invaluable contributions to the success of this project. I would also like to thank our project funders. Thank you for attending my talk. If you're interested in trying out GoldRush, feel free to check it out at github.com/bcgsc/GoldRush or use the QR code. If you're interested in the stages of GoldRush, feel free to check out our posters at HiTSeq. I've also listed other talks and posters from the Bureau Lab if you're interested in our other ongoing projects. Thank you once again.